Okay, so I formally welcome Professor Gautam Menon for this talk, and we are thankful that you accepted our invitation and you agreed for this wonderful talk. We are sure that we will be greatly benefited by your talk. So let me talk about the department formally. So since 1964, when BITS came into existence as a deemed university, it existed even before 1964, but as a different colleges. But since 1964, it came into existence as a deemed university. And from that inception, physics department is an integral part of BITS Pilani. So currently we have 20 faculty members who worked in different areas like soft condensed matter physics, Naveen Singh and Manjula Devi and others also, and particle physics also, and nuclear physics, and cosmology, astrophysics, atomic and molecular physics. So many different areas we work, both in the experimental work and theoretical work. And currently we have around 30 students, PhD students. And for our MSc program, normally nowadays intake is around 75 to 80. And that's all from the department side. So I invite Professor Naveen Singh to say a few words about you, and then you can proceed further.
Thank you, Rakesh. I hope I am visible and audible. So I formally welcome Professor Gautam. In 2017, in DNA physics workshop, I invited him, and he was finalized, but somehow at the last moment he said he cannot come. So after 4 years, online platform, he is here. We are thankful to him. Professor Gautam did PhD from Institute of Science, Bangalore. And after doing several postdoc, he joined Institute of Mathematical Science, where he is professor right now. He is additionally adjunct professor of TFR Mumbai, and also right now he is acting as a professor at Ashoka University. He has worked remarkably he has contributed, I should say, not worked, he has contributed in the area of condensed matter, especially soft condensed matter, biological physics area. He is one of the leading scientists, and not only in India, but in the world in the area of biological physics, theoretical biological physics. He has a big group at Math Science. He is also a fellow of National Academy of Science, and he's very active on social media, in which he discusses many things about the biological physics and how it impacts society. Right now, he's actively working on how COVID spread in the society. That is a very, very remarkable work he is doing right now. So without coming in between the audience and speaker, I invite formally Professor Menon to start his talk.
Thank you, Naveen. Thank you, Rajesh, for this very kind invitation. Let me present my talk. I'm very happy to be here at least virtually at BITS Pilani. And I've always wanted to come there, and hopefully someday when we're over COVID, it will be possible for me to make a physical visit. I look forward to that day. Let me present my talk, and can you just, if one of you would confirm that you can see my screen and that you can hear me clearly?
Yes, no problem.
Okay, thank you.
Yeah, it is okay.
Thanks. As was mentioned by Naveen, I'm currently at the Institute of Mathematical Sciences and at Ashoka University. So my work for many years was confined to statistical physics, soft matter physics, and biophysics. I started off as a condensed matter physicist, working at the interface of condensed matter and statistical mechanics. But nowadays, I'm really a biophysicist, computational biologist, and as was mentioned earlier, the prime interest of mine at this moment is the modeling of infectious diseases. And much of this is intended to help government decide what is its policy across different states of the country, And as all of you know, I'm sure that even in Pilani, you're feeling the pressure of the slowly increasing number of, not slowly, rapidly increasing number of cases that we're seeing across India. So to try and understand that from some sort of mathematical, epidemiological, modeling type of point of view, it's possible to provide input to various agencies of the state to try and see how we can best prepare. And finally, we would like to do what is best for our country at a particular time. And that's been my interest much more recently. So I made the shift between thinking about really physics problems to problems at the interface of physics and biology in 2012, roughly 8 or 9 years ago. So my work has centered around using the tools and training from physics, all of my training in physics through my undergrad studies, my graduate studies, my PhD, my postdoc, to now understanding specific biological problems from a quantitative perspective. And that, I think, is the whole point of doing biophysics. It's not just to create interesting models for systems, but to be able to compare directly and quantitatively between what is seen in the experimental situation and what we can put into our models. So one question that has really motivated me for the last many years is this question of, can you bring to biophysical modeling the sort of rigor and precision of models in physics? And there are some reasons why you might think this is possible. There are some reasons why you might think this is impossible. And I will argue that it's really in the nature of the questions that you ask that you might hope to be able to make progress on this particular direction. So here's an example of what I mean by a quantitative agreement of theory and experiment. So this is the universal background, microwave background, and you can see the data points and the fit to the curve. The fit to the curve is the fit to the black body curve, which you can see below, of the intensity d specific wavelength per unit volume. And you can see that this is pretty much a perfect fit. In fact, it's such a good fit that the error bars on each one of those points that I have shown, each one of the black squares that I have shown, is 100th the size of the symbol that is used to plot it. And I think often people from other fields don't realize how exquisitely sensitive and precise experiments in physics can be. Right now, we're talking about experiments on the G-2 factor of the muon, which show very, very small discrepancy in something like the 8th or the 9th decimal place. And that's the accuracy with which we can now, to which we know that our arguments about physics and our understanding of physics is complete. I'd like to compare this to a typical experiment in biology, and you can see that the spread of the data points between the black data points and the blue and the open circles. And this is just probably under 2 different conditions. I haven't shown you what's on the x-axis, I haven't shown you on the y-axis, but it helps to make a couple of points. First of all, biological systems are intrinsically very noisy. Unlike this data here, where the noise in terms of the error bar was really smaller than the point that I used to plot it, which is an amazing statement that one can make. Over here, the error bars are all over the place. You can look at the spread of the data, and to make statements about how the mean is shifting from one condition to the other is really a statement that relies on probability. We can only say that with some probability, these are different, with some probability, they're likely to be the same. And it's nowhere like the sort of precision that we had in the earlier experiment. There are other things that make biological systems interesting from the point of view of a soft matter physicist. These are small systems, they're soft and deformable, they're confined, almost always they're confined, they're crowded, and they're out of equilibrium. So I want to take some of these keywords, small, soft, deformable, confined, crowded, out of equilibrium, and to illustrate how they turn up in 2 prototypical problems that I'm interested in, that I've been interested in over the last few years. So it's these systems that I want to describe to you. And to do this, let me just sort of say one thing at the outset, that we will be talking about cells. Cells are the fundamental unit of structure and function in anything that is living. And all cells arise from pre-existing cells through a cell division. So this is the cell doctrine or the cell theory. And this is going to be fundamental to this is going to be the one thing that I will ask you, the 1 or 2 things that I will ask you to remember from your high school biology lessons. The first point that I want to tell you about is a problem called axonal transport. Okay, so I will explain each of these terms and tell you what they are. But let's first go from that picture of the fact that cells are the basic unit of living things to asking about how many different types of cells are there in your body. Right now, we know there are about more than 230 different types of cells, but they're all organized, even though their shapes are different, their types are different, what they do are different. Your skin cells are different from your liver cells, are different from your kidney cells. The broad organizing principles are the same in all of these different types of cells. So a muscle cell might look like you in the top left-hand corner, the blood cells look like that, the sperm cells look very different with a long tail, skin cells have this more square appearance. But the cell that I'm going to be interested in for the later part of the talk is a nerve cell, a neuron. But right now, I just want you to remember that there's a whole range of different types of cells, but they all kind of look the same from a functional point of view. The organizing principles are the same, even though their detailed shapes may differ. So in this problem, the first problem that I want to tell you about is the problem of the nerve cell or the neuron. The neuron is another word for a nerve cell. You can see that it has a very different appearance from the other cells. It looks much more pointed. It has these little things at the ends, at the tips of these little pointing regions that it has here. And the important thing that we need to know about nerve cells is that they have these long extended structures, which are called axons, and these terminate in what are called synapses. So 2 words, neuron for a nerve cell, axons for the extension of the nerve cell. All of these are shown in the picture below, and the axon terminates in a synapse. So the synapse is what connects nerve cells to nerve cells. You have the axon coming, touching, as you can see, the second cell touching the third cell. And it's this relay of different cells and the chemical messages that pass, chemical and electrical messages that pass from one to the other, that is responsible for nerve conduction, for example. It's responsible for how your brain processes information. All of this really comes back to the biological feature that these cells talk to each other through the points at which one interacts the other. This point is a special point called the synapse. The synapse is loaded with very interesting molecules. These are called neurotransmitters. And these neurotransmitters are released across the gap that separates one cell from the other. This is called the synaptic cleft. And you have a whole bunch of molecules there. And when the cell is stimulated in some way, typically it transmits an electrical impulse. That electrical impulse leads to the release of these molecules from the edge of the cell. This goes and stimulates the cell that is on the receiving end through a part called the dendrite. So the interaction between the synapse and the dendrite leads to one cell communicating information. For example, excitation of one cell is being communicated to the second cell. So whenever biologists talk about biological experiments, they always have in mind some particular model system. And often people choose very specific model systems to discuss specific questions. The model system that my collaborators use is something called C. elegans. And this is a worm, it's a round worm. That's what it looks like. It's a few millimeters in size. You can see it easily under a microscope. It's transparent. It has a bunch of advantages that make it easy to study, including the fact that its genetic code is completely known. But that's what the worm looks like. And this is going to be the experimental system which I will choose to answer a certain set of experimental questions and then try to relate that to a theoretical modeling. So as you saw, the C. elegans, the worm C. elegans is transparent. And in addition, because we know so much about genetics nowadays, we can change the genetics of the C. elegans in various ways that make it interesting for us. We can look for specific mutations in the genes that it contains that lead to different types of behavior. What's interesting to us right now is that we know all of the nerve cells and the programs by which they're connected inside this. So this is just a picture that shows you all of the different nerve cells inside the C. elegans worm. Right now, C. elegans totally has 302 neurons out of 959 cells. So this is what makes this particular part of biology so very quantitative. You know exactly the number of cells that the adult organism has. You know what fraction of those cells are neurons, are nerve cells, and you know where they figure in the organism. So there are 302 neurons, and these connections between these neurons, there are 7,000 of these connections, and these have all been mapped out. We know exactly what they're doing in the adult C. elegans worm. Just as a comparison, the human brain has 86 billion neurons, that's 86 into 10 to the power 9 neurons, and 100 trillion synapses, that is points at which nerve cells connect to each other. So that should give you some idea of the scale at which we are thinking about how does human thinking and human intelligence and human capability differ from those of lower organisms. So one is just a scaling up of the total number of neurons or nerve cells that one contains from 302 to 86 billion. So we're going to be interested in one very particular nerve cell, and this is called the touch neuron inside here. So one of these nerve cells that you can see in that picture there is called the touch neuron. And here is that neuron sort of expanded out to give you an idea of what its structure consists of. So this is the cell, this is the nerve cell. As I said, the nerve cell has a long extension. It's called the axon. And stuff is going from there to the end of the axon, which contains the synapse. As I said, the synapse is full of chemicals, and these chemicals have to reach there. Question is, how do they reach there? So the way they reach there is not by simple diffusion, but because they're packed into a bag called a vesicle, and then they're transported by very small, in a sense, biologically mechanical objects, which move them. These are called molecular motors. And molecular motors are responsible for moving material across cells, especially the cells in our body. So we will call this cargo. Anything that is inside a vesicle that is being carried by a molecular motor, we will call cargo. There are many types of cargoes depending upon what has to be delivered to different types of cells, but they all move along these specific extensions of these long rod-like objects called microtubules. So here is the bottom line. Cells move, especially the large cells, not the small bacterial cells, but cells move material back and forth inside by putting them onto molecular motors. These molecular motors are responsible for the transport of cargo from one point to the other. And these molecular motors move along long, rigid polymers called microtubules. I'll come to some more examples so that it's a little clearer here, but just keep these words in mind. So here's a movie that you can see of what microtubules look like. They're hollow cylinders, nanometer-sized hollow cylinders. This shows a microtubule forming out of many monomers coming and attaching at the end of this. And you can see how microtubules disassemble. This is how they come apart. So these are molecules that are constantly forming and breaking apart, forming and breaking apart. And as they break apart, they release their monomers, but these monomers can go and join at the end of some other microtubule. So here is the interesting thing. This shows a vesicle, which is that big blue bag that you can see at the end. And that little tail-like thing that you can see, the little low structure that you can see, is a molecular motor. So the molecular motor is, as I said, you should think about it as a little motor that moves forward, holding this big bag. It's just like a porter that you might see in Jaipur station or somewhere, wherever you might want to look at. So this is the microtubules, the cargo that is being carried on the microtubules, and the motors responsible for carrying that cargo. So we know a lot about the biology of the cargo. The biology is contained. So these vesicles are called synaptic vesicles because they're intended for the synapse. We know a lot about the motor, the different parts that go into the motor. You can see that on the left-hand side, where the different motors, different things that are important biologically can be labeled if you wanted to study them under a fluorescence microscope. And the picture to the right is a picture of the motor carrying cargo moving along special sites on the microtubule. These sites are spaced by about 8 to 9 nanometers between them. And therefore, the motor takes very precise steps as it's moving. And these are the steps that are illustrated over there. In order for a motor to move, it must consume energy. It requires energy to be able to move because it's in a very dissipative fluid environment. And anyone who works in soft matter will know the difficulties of working with things that are very, very small because they're constantly moving through Brownian motion. So for something to move in a targeted manner from one point to another, it needs to be able to circumvent that Brownian motion, and you have to supply it energy in order to be able to do so. And that energy comes to it in the form of an ATP molecule. So let me summarize what we know so far, just so that you can understand the bigger picture. We're going to be thinking about worms, the C. elegans worm that you can see on the top left-hand side. We now know what the structure of that worm is. We know where the neurons inside that worm is. And some of these neurons are of specific interest to us because we can study them. This an expanded picture of the neuron looks like this. It has a long part to it, which is the axon. And this axon terminates in axon terminals at which you have synapses. And these help neurons connect to other neurons. The cell, of course, takes in input from many other points over here. So from the dendrites, it takes an input. And then it transfers that input from one neuron to the other by releasing chemicals at the end of the axon at what we call the synapse. All of these chemicals are transported back and forth by packing them into bags, which are called secretory vesicles or presynaptic vesicles. These bags are then transported by molecular motors. There are two types of molecular motors. Some move in this direction, some move in this direction. So they're retrograde and antrograde molecular motors. And these are what are really responsible at the core for all of biological transport in this animal or in this neuron that you can see. A little more detail, we sort of imagine this. We looked at the scale of the vesicle compared to the scale of the motor. The motor is itself about 80 nanometers in size, very small from sort of top to bottom. And you can see in the picture below, you can see the motor moving, carrying its cargo. So that's all I've described so far. And this has been a fair amount of detail if you haven't come across it so far. But I will just ask you to think of something that is carrying a cargo moving in one direction. And that's the motor carrying cargo. And that's going to be a fundamental interest to understand what is the nature of this transport inside this complicated milieu. How exactly is cargo transferred from the cell body where the cargo is made and put into these bags to right up to the end of the axon where it is going to be released at the synaptic tract. So here's an example of what happens if you put a fluorescent probe on the cargo in this particular case that's being transported. So you can see that some cargo is moving to the right. These little bright dots are the fluorescent probes. You can see something moving back and forth, something sticking for a while. Its movement is a little bit regular. Some of them move fast. Some of them tend to get stuck in these little clusters. And these are the sort of experiments that we might want to try and understand. We want to try and understand what is the biophysical logic behind these experiments and what makes it function. So notice that motion is bidirectional. You have stuff moving here and stuff moving here. Notice that cargo can get stuck, that middle little pieces of cargo which is not moving anywhere. They're often stuck in very specific places along the axon. Even if something gets unstuck, something can come from there and get stuck again at that point. After a while, even these stuck regions tend to dissolve. Things seem to move away from these stuck regions. And if you look at the amount of time that you have stuck cargo at particular places, these can be fairly long. So there's a broad distribution of times. So remember the word cargo. Cargo is what is being moved back and forth. Cargo is what is contained in the vesicle that is being transported by the molecular motor. And it's from experiments such as this. You cannot do this in a more precise level. If you want to study the in vivo, the living system, you're always constrained to look at data like this and to try and extract and understand information that comes from this. Couple of points. So biological environments are usually very densely crowded. There are many, many different types. By types, I mean shapes, sizes, chemical properties, etc. of molecules. So that's a sort of picture of the level of crowding that exists in a typical cell. And we know what crowding means. The picture at the bottom right is, of course, the crowding. So probably a Bombay local train is what I would guess. But maybe this is even too crowded for that. It may even be a long-distance train. So we do have an idea in India what crowding means. So we can try and transplant that to thinking about crowding in a cellular context at this level. The general question that I want to address is how does a cell deal with efficiently transporting material in a crowded environment? And as a biophysicist interested in these broader questions, the question that I want to answer is what are the general principles that may be helpful in understanding how transport happens within cells, and what could these principles actually be? I want to draw an analogy between multiple kind of similar situations. And I will argue that the similarity between these are all traffic problems. So we started with motor protein transport, the motors that transport cargo within the axon. The picture in the middle is cell migration. So cells are moving along specially designed tracks. So you can see that they also move very purposefully along these things. And of course, vehicular traffic, each car is typically about 2 meters in size. And you can look at the kiosk there in a typical Indian road pre-lockdown, what it actually looks like. So look at the commonalities between these. And I want to suggest that the commonalities between all of this is that they're all traffic problems at their root. What do we know about traffic? As you can see from the earlier examples, traffic encounters obstacles. There are many blocks that it might encounter when it tries to move along the road. There are breaks in the road. Here's an example of a multiple break after an earthquake, where the road has just caved in at some point. So any car or vehicle that is trying to move from this point to that point will encounter these breaks and will have to stop at these breaks. There's, of course, multiple contexts in which you can think of crowding in traffic jams. And this sort of crowding may be related to the crowding that you expect inside a cellular context, where you have many molecules of many different types coming together. And in that, it is in that sort of milieu, it's in that crowded environment that you might expect this motor protein to actually move around. And of course, there's also crowding from other motor proteins carrying cargo, just as there's crowding from these multiple types of cars and vehicles that are actually blocking it. So I want to take these examples, these multiple examples of traffic that I showed you, and translate these into what might be happening at the level of the cell in order to understand the experiments that I showed you about. So as in the experiments, we know that traffic is bidirectional. You have cars going here and cars going here, or vesicles going here and vesicles going here. They can change track. You saw that the microtubule had these multiple tracks. There are 13 what are called protofilaments that make up the hollow core of the microtubule. We can take many parameters about how vesicles move from the experiments and use them in the model to benchmark the model. And then, of course, it's just a question of taking this and making a simulation out of it. So let me show you what the simulation data look like. Here is a simulation where we've taken one microtubule, and we're looking at how you traffic back and forth within that microtubule. So each of these little dots that you can see that are moving back and forth are vesicles that are being carried by molecular motors. You can see that there are certain regions where they stop, and these regions are typically the ends of microtubules. They're also associated with large, slow-moving cargo. Remember that I said there's a whole lot of different stuff that is moving back and forth. The large cargo is a mitochondrion, which moves very slowly. You can see a whole traffic jam develop near in the background of that mitochondrion, but then things are still available to move. They can sidestep and they can move across. We know there are multiple states of this cargo. We can put that into the simulation that we drew. There are 2 states of anterograde motion, that is motion towards the synapse, and 1 retrograde motion state, that is motion away from the synapse. So this is our idealization and from the point of view of simulations of what was being seen in the experiments. So now I can get a little more quantitative, take data like that and understand what are called experimental chymographs. So a chymograph is something that plots time in the vertical direction and then plots trajectories in the horizontal direction. So any line that is at an angle here is something that is moving with some velocity either forward or backward. Any straight line in the chymograph, you can see a couple of 2 dark straight lines there. That refers to something that is stationary, whose position does not change. So you can construct simulation chymographs, you can study experimental chymographs, and then you can attempt to compare them to see to what extent is the simulation reproducing what is there in the experiment. So here is a complicated simulation chymograph, and you can also look at this in more detail, little parts of it, and see are the different types of changes in the motion of vesicles that I can see in the experiments, are they reproduced in my simulation? So here's a bunch of different, the dispersion, mobilization, formation, stopping, the stationary cluster formation, crossovers, etc. All of these one can see examples for in the experiment as well as in the simulation. So now we can be fairly sure that the simulation is being able to capture what is there in the experiments. Now we can begin to look at really the phenomenon that we were interested in. And that phenomenon was the fact that if you looked at these simulations, if you looked at the experiments, there were these regions where vesicles seemed to crowd together, seemed to be there for long periods of time. So we call them stationary clusters. And the fact that some of them could be there for long periods of time, typically exceeding 1 to 5 minutes in some cases, we said that these were long-lived stationary clusters and they formed permanent blocks. Okay. So now you can see the experimental data for a particular label that we have, a RAP3 label. And then we can say that what do we get in simulations where we have these multiple motion states of vesicles, and we can say, can we relate what is seen in experiments to what is seen in simulation? And the answer is yes. We can now test multiple applications of this to check that it actually works. After that, with this confidence, we can do what is called a bleach recovery experiment. So what is done there is to irradiate a certain region with high intensity laser light, and essentially all the fluorescent molecules there cease to fluoresce for a certain amount of time. And this is, you have now bleached that particular region, and the unbleached region outside is where you have a fresh influx of molecules, and these will be molecules such as motors carrying cargo. And then you can ask, from the way this bleached region is gradually filled in, about the properties of the stuff that is being transported inside, about the vesicles as well as the molecular motors. And you can see, if you change the vesicle number, if you change the time over which you bleach, you can see that exactly the same bleach recovery is happening. As you can see, it's an experimental plot on the top right-hand side, and these are theoretical plots on the bottom right-hand side, and you can establish a one-to-one relationship between what you see in the experiments and what you see in the simulations. So with all of this, we can now begin to answer the questions. Where do vesicles cluster along the axon? Where do these clusters actually happen? We can answer that these are often in regions that are blocked in some way, typically the ends of microtubules or breaks in the microtubules, or regions where another protein called actin is present in high density, other types of cargo such as mitochondria, but they also happen elsewhere. And they happen elsewhere because something moving like this and something moving like this on the same filament or on the same traffic road can come and meet each other, and then neither can move apart. You have to wait until something can sidestep to the right, take another lane, or decide to move backwards so that to give space for this guy to move forward. Only then will you unblock. But having clusters that arise purely naturally, even away from these special pieces where the road has broken down, is completely normal. We can ask, is the formation of these traffic jams where things come together and then don't move, is it a property only of diseased neurons? The answer is no. Even across multiple types of healthy neurons, you can see exactly the same type of behavior. Once you stop cargo, once things come together, they stop, you have a cluster of cargo there, can it move again? Can it come apart again? The answer is yes. Typically, these are very dynamic. They contain 1 to 4 vesicles, but these vesicles keep coming and going at a steady rate. So you don't tend to build up these permanent long-scale traffic jams. You may have heard of China, where you had a 3-day traffic jam. It took 3 days to resolve that. You don't have these large-scale, very long-lived traffic jams building up. They tend to be able to dissolve. So this is what is important to this is the ability to reverse completely for a vesicle that is coming like this to decide to go backward. And then this is an interesting point. Why is it that permanent traffic jams don't form? And now we have been examining that particular question. Reversals turn out to be important, as also the fact that these are dynamic clusters. The last sort of interesting point, or the second last interesting point, is that such jam regions should be very crowded. There are many vesicles jammed in together. What does this imply? And it's particularly important because the axon can be very, very, very long in comparison to the rest of the cell, the body of the cell. So the idea here is an interesting idea, that the role of stationary clusters that can form randomly anywhere is basically to hold and to release vesicles. These are dynamical reservoirs of vesicles. And the idea of the dynamic reservoir is an interesting idea that comes from thinking, from a physicist and a biologist thinking together about this particular problem. But here's where it's useful, and this is where an interesting insight comes in. Remember that I said that the axon is very long. Those pictures were an exaggeration. The axon really looks like this. You have the cell body, and then you have this long axon that can be very, very long. It's equivalent to thousands of loops, as you can see here. And right at the end of that is where the synapse is. So the idea is that the clusters, the stationary clusters, are a way of storing cargo along the way in what we call dynamical reservoirs. These are dynamical because they can dissolve completely, they can reform completely, they can form and dissolve at any point along the axon. But they all have this property that they can hold 4 to 6 new vesicles at a time, and then after some time release, reform, release, etc., etc., etc. This is important because of the following idea. Suppose I had to move something from Bombay to Chennai. Let's say it was vaccines from Bombay to Chennai in the current context. Now Chennai has run out of vaccine. What should Chennai do? Should Chennai wait for Bombay to send Chennai vaccine, in which case it will take 3 days to reach there by road? Or the alternative is that Bombay keeps on sending Chennai vaccines, but these vaccines are not continuously streaming in through, they're held at various points in between. So it might stop at Nagpur, it might stop in Hyderabad, it might stop in Tirupati, it might stop in Chennai. So there are these go-downs along the way, where you put in vaccines, hold them for some time, release them, put them here, hold them, release them, until they're finally received in Chennai. The advantage is that if Chennai suddenly runs out of vaccine, it doesn't have to refer all the way to Bombay to get them. It just needs to send a signal to the nearest reservoir so that they can be released and the vaccines can be taken inside. The second problem that I want to tell you about in about 20 minutes is a problem called nuclear architecture. So that was one problem. I'm going to tell you about 2 biophysical problems that I've been interested in and the broader information that we can get about these problems. So here the question is, what determines what is called large-scale nuclear architecture in human cells, and how does one think about this as a problem in physics and, of course, in biology? The word that I would like you to take from this part of the talk is the word called chromatin. And chromatin is the material out of which chromosomes are made of. So you know that your body contains cells, your cells contain nuclei, your nuclei contains DNA, but the DNA is not just present as a free molecule floating around, it's tightly packaged into something called a chromosome. And the material of the chromosome, the DNA and all the stuff that binds to the DNA is together called chromatin. So chromatin is compacted DNA. So you can see how the chromosome there becomes a compacted DNA. And at even smaller scale, that reduces to the standard DNA double helix that Watson and Crick told us the structure for. Okay. The chromosomes that contain the DNA contain genes as well as regions that are devoid of genes. So the important information, the information contained in the genes that is intended to be written out, is spread out along all of the DNA that is contained in chromosomes. That's the information that I would like you to remember. I want to tell you about the old picture of chromatin and the current picture of chromatin. The old classic picture of chromatin from about 1990 and before 1990 was that the nucleus really had this very unstructured chromatin. These are all long molecules. These molecules were all mixed up together like a dish of spaghetti or Maggi noodles. There's no difference. There's no structure to the way these molecules are arranged. We know something very different now. We know that chromosomes are what are called territorial. They occupy very specific regions within the nucleus, and the locations of these regions are not random. They're distributed according to some probability distribution. I'll talk about human cells in what is called interface, between cell division, because that is for us a simpler problem. The last ingredient that we need to think about this is the physical context. And the physical context, as I said, is a context in which the cell is out of equilibrium. We saw one example earlier where I said that the molecule, the molecular motor, had to take in and had to constantly consume energy. That energy reaches it in the form of an ATP molecule. So that's a little cartoon that you can see here. That's the molecular motor with 2 heads. It grabs an ATP molecule, uses the ATP molecule, uses the energy contained in breaking, when it breaks the bond, the ATP bond into ADP, adenosine triphosphate into adenosine diphosphate, and uses the energy gained from breaking that bond in moving forward. So after that, it releases the ADP, and the ADP goes somewhere else and is reconstituted into ATP. So that's the way in which the cell has energy to play around with at the microscopic level. And by keeping the concentrations of ATP and ADP out of equilibrium, the cell manages to be out of equilibrium and to have a constant energy supply for anything that it requires. This process is called ATP hydrolysis. Energy input happens at the nanoscale in biological systems as a consequence of ATP hydrolysis. This idea is called activity. The fact of the way in which biological systems are out of equilibrium is called activity. These are active systems, and chromatin should be thought of as active soft matter. Just as my earlier example, I said we're going to look at confined systems, we're going to look at non-equivalent systems, we're going to look at soft and deformable systems. All of that there together is in this particular example. So the challenge is, I give you 1,000 nuclei, you label the chromosomes within that nuclei. From that, you construct a distribution function. How far is the center of mass of that particular chromosome away from the center of the nucleus? How far is the DNA contained in that chromosome away from the nucleus? And I construct a certain statistical representation that you can see on the bottom right of this curve. And now the question is, how do I understand that from some sort of modeling point of view? So as I said, let's just remind ourselves that the physical context is that any living system at the nanoscale is constantly consuming energy. This energy is being fed to it at a very local scale. This locality of energy, of non-equivalent energy supply, is called activity. And now my hypothesis is going to be that it's this activity that is not the same in regions which contain genes versus regions that don't contain genes, that is responsible for all of the intrinsic properties, all of what is interesting about large-scale nuclear architecture. So here's how I'm going to model this. A chromosome is made up of bases of DNA. I'm going to group those units into one megabase, so 10 to the 6 units together. And I'm going to treat this as a monomer. And out of these monomers, I'm going to build up a chromosome. Because I know the human genome, I know exactly which genes are where in which particular type of cell. And I know which genes are turned on and off, so I know about the activity of those genes. So the fact that there are different gene densities in these monomers is information that I already have. And these basic units, the 1 MB units, not the fact that I don't have to worry about really the microscale single base pair, is because these are known to be building blocks of chromosome territories. And then the activity of each monomer, how active it is going to be set by how active the genes are that it contains, that particular monomer contains. So here's where the physics enters. The more activity there is, the larger fluctuation you expect to see, because all of this is energy consuming. Any motor that is trying to press on a filament over here will exert a counter force on the reaction force on the environment that surrounds it. The sum of these many reaction forces from the uncoordinated motion of molecular motors is like a thermal noise, but this thermal noise can be much larger than the type of noise that comes from purely thermal fluctuations. So this is an unusual polymer. It is a polymer in which some regions are hot and some regions are cold. The hot region is where we map onto where the genes are, most of the genes are. The cold regions we map to where the genes are not. So there are gene poor regions and gene rich regions. And this, we know all of this information because we know the human genome. We can map it onto a problem of just simulating a polymer of this form. So we have a bunch of simple models here. The simplest model is just to look at the gene density across each of these segments. Just say that the highest gene density monomers, I'm going to promote them to having a higher temperature. Everything else I'm going to keep at a standard physiological or body temperature of about 38 degrees centigrade. And now I'm going to simulate this model. This is not real temperature, I again emphasize. This is the temperature associated with the sort of mechanical fluctuations that come at a purely local level. And these mechanical fluctuations come because the cell locally is consuming energy to do something very specific at the level of its DNA. And we now know exactly what it's doing in most of these biological contexts. Because we're simulating individual chromosomes, we're simulating every different chromosome inside the cell nucleus at this level. We can look at different shapes of the different nuclei. Some nuclei tend to be more flattened, some tend to be more pulled out, some nuclei tend to be spherical. We can look at the situation in each one of these. Because we're actually simulating the experimental system, I know which is my chromosome 18, which is my chromosome 19, and then I ask about differences between them. So one important feature that we know about cells is that the gene-rich regions tend to be more inside, the gene-poor regions tend to be more outside. So I can ask exactly this question, given a large number of simulation configurations, where are the genes sitting? Are the genes on the inside or the genes on the outside? So that's a picture that comes from simulation, where you can see that the light-colored part is an excess of genes, the dark-colored part is a decrease in the average number of genes. So you can see that most genes tend to be concentrated towards the center, and there are relatively few towards the periphery, which is dark. Okay, so this is a purely theoretical prediction for what should happen. Here are experimental distribution functions. As I said, I said the statistical problem was to compute how many pieces of the DNA corresponding to a specific chromosome were present at some radial distance away from the center. This is experimental data. And now on top of this, I can superimpose my theoretical prediction. And this is how the theoretical prediction does for different chromosomes. You can see that chromosome 18 and 19 are very different from each other. They have different gene densities. Chromosome 19 has 4 times the amount of genes for its length than chromosome 18 does. You can see that chromosome 19 is more towards the inside. It peaks more towards the inside than chromosome 18. Look to the picture to the right. Whereas chromosomes 12 and 20 have very different sizes with the same gene density. You can see that their distribution doesn't change much and is reasonably captured by the theoretical arguments. How do we form? So chromosome territory, the idea that chromosomes are territorial, is an old idea. It was proposed many years ago, but it was really seen only in the last 20 years through a very beautiful technique called the fluorescence in situ hybridization. And that amounts to adding specific fluorescent labels of a specific color to specific regions of DNA inside the chromosomes. So now I can say that this region is dark, this region is light, etc. And from here I can get to distributing all of the chromosomes in an experimental picture. I can now compare this to what I get from theory and find that I can reproduce exactly the same sort of territorial structure from my simulation. This is a very robust consequence of my simulations. Finally, let me tell you about the center of mass distribution. Again, experimental data. This is the center of mass of chromosome 21, 22, 19, 20. There are totally 22 chromosomes and 2 X or Y chromosomes that a human cell has. So 23 pairs of chromosomes, totaling 46. I can say how far are they on average from the center of the nucleus. That's this plot here. And now I can do this both in the simulations and superimpose the simulation plot on top of the experimental plot. And you can see the simulations actually do very well in this particular context. You can see that they predict the slow, this gentle behavior as a function of chromosome size or where chromosome centers of mass are located with respect to the center of the nucleus. So given this, I want to sort of argue that these predictions agree very well with data, even if it's at a quantitative level. And this is not just a qualitative comparison of data between saying some of it is okay, some of it is not okay. This is a direct comparison across multiple experiments of a theoretical prediction with an experimental result. This also agrees with our broad understanding from lots of different measurements. The measurement that measured the center of mass position was different from the measurement that measured the distribution of DNA inside this, was also different from the measurement that measured territoriality. So the fact that a theoretical framework can agree with multiple different measurements is very unusual in biophysics. And the fact that you have this sort of agreement with this at a qualitative and a quantitative level is also very unusual and very interesting. So let me finish up here. This has been a short talk. I will say a few more things. But I want to tell you that I told you about 2 problems in biophysics. I said that I'm a physicist who comes from a fairly traditional background in thinking about condensed matter physics. But these problems in biology are important problems that people have just begun to think about in a physics context. So the problem of large-scale nuclear architecture, what determines where chromosomes at the 1 MB level and above are distributed? How are they distributed? What are the rules? What are the physical rules or the biophysical rules that determine where they are? This is something that this particular work of ours addressed. The second question was, how do cells transport material from inside? So that was a cellular transport question that was first in the sequence in which I did it. We looked at a prototypical example of cellular transport of molecular motors transporting cargo that was encapsulated in vesicles, taking them from one point to the other. And we said, what do we need to know to understand the details of that particular physical process? We said we needed to account for the fact that you had bidirectional motion, that this motion was on filaments, that these could encounter with each other. But we also said you had to put all of the complexity that existed in the biological system, the fact the broken roads, which meant the broken microtubules, other types of slow-moving cargo. All of you who've been stuck behind a very slow-moving bus going uphill or a lorry going uphill will know what it feels like. You can see the whole line of cars behind you, just trying to get ahead, trying to slip past that bus that is moving so slowly. So there are analogies that I will draw here between systems as we understand it from our current experience of traffic and the ability of to be able to take that intuition derived from that and translate it into a model of this sort. So let me just talk about very briefly, I said this was the talk about interdisciplinarity. So what are the points that I want you to take away from this discussion so far? What are the lessons that we can learn for thinking about interdisciplinarity? The first point that I want to make is that the future of science, not the current state of science, but where science is going, in fact, where it is also currently, is increasingly interdisciplinary. And many of you are teachers, some of you are students. I expect that it's important for both educators as well as students to understand this. and to provide students with tools and for students to be able to imbibe those tools that enable them to move from one type of problem to another type of problem. There's good interdisciplinarity and there's bad interdisciplinarity. Some interdisciplinarity is just boring. It just says, let me try this tool from here and try it out here. That may not necessarily be boring, so that word boring is a bit too strong, but it's a fairly standard thing that doesn't really need two people from different areas coming together to discuss the problems and how one method from one area might solve a problem in another area. And this is back and forth that I think is very important, especially when we talk about between physics and biology, physics and chemistry, chemistry and biology. These are all interaction fields or overlaps, which are very important for at least the next several decades, and we need to be able to prepare for that. What I'd like students to take away, and faculty also to take away, is the need for new tools. Most advances in biology, most advances in science have not really occurred because some genius thought of a new way to do things. They come from the development of new experimental tools, new experiments that can push existing technology in a further direction, which is why many Nobel Prizes nowadays are being given just for the development of tools that enable you to look at cells, for example, in more detail through better microscopy methods, or a bunch of other tools. The tools for being able to label cells with particular fluorescent molecules was another Nobel Prize in chemistry. So there are both experimental and computational aspects to the overlap between different fields. Often you can take an experimental field and put a computational technique onto it. The new tools can be computational, a new way of thinking about a computational problem. In the examples that I gave you, we made many approximations to extract from that the crucial biophysical intuition that was relevant to the study of those problems. A general point is that interesting problems may lie outside your area, but you may be skilled to address them. You have to keep your mind open. And you have to be able to, as a scientist, some of you will become scientists in the future, I'm sure the younger people among you. You have to be able to say that I'm going to think about problems, but I'm going to understand what is it that I'm in a good position to contribute to those problems. And I'm going to see how can I develop the right sorts of interdisciplinary collaborations that might enable me to answer them. The last thing I want to point out is the importance of analogies. We made a bunch of analogies between traffic and motion inside cells, etc., etc. There was no simple analogy that existed for the nucleus, for chromosomes inside the nucleus, but we made it a model in a very particular sense. We said, let's extract from this what we believe to be important. Let's ignore what we think is not important. And then we can ask if what is important is enough to reproduce the features that are crucial to the biological system. That's the importance of analogies, I think, is a very fundamental issue that anyone thinking about being interdisciplinary should consider. I will leave you with this article. This is an article that appeared in India Bioscience in November of 2018. That is the link for you. So just search on India Bioscience for Gautam Menon. You will probably find this article. And this describes some strategies for making interdisciplinary collaborations work. So this may interest you in the context of what I've spoken to you so far about. Let me finish up with acknowledging all of my collaborators. The principal collaborator on all of the work that I described to you that was about transport inside cells was Sandhya Kaushika at the NCBS Bangalore and later the TIFR Mumbai. These are the wonderful students who have worked on that project, Amrita, Kausalya, Parul and Kirtana. On the left are the people who work on the theoretical side and the computational side, Reshma and Vinod Kumar, who are my students and my project assistant at the Institute of Mathematical Sciences. The second part of the talk was by work done by Nirmalendu Ganai, who was a PhD student at that time, Shurjit Sen Gupta of the TCS Hyderabad, and Ankit Agarwal, my own student, who really developed this into an extremely detailed and remarkable tool for thinking about problems of large-scale nuclear architecture. The last thing I want to point out is Shurjit Sen Gupta was a very close friend of mine. He died tragically about a month ago. And so this talk is in his memory as well. Thank you so much. I'm happy to answer questions about anything that I've said so far. In fact, about anything that I work on. If you're interested in my work on infectious diseases, I'm happy to talk about that as well. But let me bring this particular talk to an end and thank my host for this very kind invitation to speak to all of you.
Thank you, Professor Gautam, for this nice talk. Yes, if you have any query, you please raise your hand so that I can pass your query to the speaker. Yes, Professor Vikram.
Yeah, I had two questions. One is, what was that unit of chromosome size which you used?
So what we use is we grouped each, the smallest size that makes sense for a DNA helix, double helix is the base pair. That's the sort of elemental unit that we understand. We group them in units of a million, of 10 to the 6 base pairs, and that defined for us our basic monomer. So totally the simulation has 6,400 monomers odd because we're working with 23 pairs female cells, 23 pairs of chromosomes. And if you break the human genome of 10 to the 9 base pairs, roughly 3 billion base pairs, that's the number that you will actually get.
So I'd just like to ask one more question.
Yeah.
So when we are talking about, let's say, artificial neural networks,
yeah,
artificial neural networks use only very bare minimal features from the natural neural networks.
Yeah.
So do you think there is any other feature of natural networks, if it's incorporated into artificial neural networks, will like lead to a lot of developments in the field of artificial neural networks?
At this point, I can't think of anything. I think the question, of course, with neural networks is how do you get the sort of collective behavior that you see that might represent, for example, the ability of a neuron to generalize from, of a system of neurons to generalize from past experience in a particular way. So given that this has been so intensively studied, I can't automatically think of something that has been missed in these studies, but is possibly of crucial importance right now. And I would guess no. I would think maybe we have not understood something fundamental about the nature of computation, but that may be more on the theoretical end rather than the experimental end. I think experimentally we have as much information as we need. That would be my tentative answer, but I could be wrong about that.
Thank you.
Yes, Amrit Kumar.
Please go ahead.
Yeah, hello, good evening, sir.
Good evening.
Sir, I want to ask a question. That when you start theoretically modeling infectious disease bacteria or virus, so sir, let's talk about COVID. So firstly, for COVID, we didn't know anything about that virus, right? So what are the challenges we face about the modeling of a particular bacteria and virus? And I'm talking about, I usually work in sensing applications of different sensing applications of these viruses, and I try to make a device out of it to sense these types of bacteria. So talking about the theoretical aspect, so what are the challenges we face when we try to model these kind of bacteria in which we don't know anything, we don't have any prerequisite knowledge about those viruses and bacteria. So what are the challenges we face, sir?
Okay, so let me make one slight sort of modification of that. We do know something about this particular COVID-19. Because COVID-19 is very related to the original SARS coronavirus. So in fact, we know the structure of that. We know that which is one reason why the genome of the SARS-CoV-2 was obtained so fast. But on this, our second point is that's not an area I work in. I work in the epidemiology of diseases, the mathematical models for how in populations disease spread. So now when you're seeing a sudden rise in cases in Maharashtra and other parts of India, what is it that we can say about when that peak will happen, what might be happening in the future? And into that, we have to put all of our knowledge, our biological and clinical knowledge about how it spreads from person to person. Now we know that COVID-19 spreads through small droplets that are contained in the air, which is why mask wearing is so important, which is why ventilation is very important, which is why crowding into small rooms you should not do that. We understand many things. So this we can put into models and ask how the disease spreads. That's a very different question from the question that you're asking. How do we know about the structure? But increasingly, we know a whole lot more about the structure. We have now very good simulations actually of all of the important parts of the virus, of the spike protein. And we can even use those simulations to find out what happens if there are mutations in that spike protein where you replace one amino acid by another. How does it lead to the opening up of the spike or the closing of the spike? Potentially, how would it change the nature of antibody binding to those spike regions? So we actually do know a lot. And we know it from a whole bunch of experiments, including very refined and beautiful cryo-electron microscopy experiments, coupled with simulations.
Oh, thank you so much, sir. Thank you.
Yes, Professor Rajkumar Gupta.
Hello, Gautam.
Hello.
It was a wonderful talk, and I really enjoyed that. I have one question. Actually, most of the biological system, they are working in, or their phenomena is happening in the water environment.
Yeah.
And water in the bulk state and the water in the confined geometry, they are completely different.
Yes.
So how do you take care of this, the presence of water molecule for, such a, at the nanometer length scale and at the bulk level, micron level?
So the very good question. So if we were worrying about how to do molecular dynamic simulations at the atomic level, we would certainly have to worry about that problem. How you treat water is very important, exactly for the reasons that you're saying. We know relatively little about the hydrophobic interaction, the structuring of water around protein surfaces, and all that would be important. But we work at a larger scale that is more biophysical. We just model this sort of the vesicle and the motor is just modeled as one object that is moving. And for that, we can take experimental data that looks at velocities, pause times, etc., and put that into the model. So we don't worry, we know that those details are there, but the only place that they enter our models is at a much larger scale, where only specifying average properties, the average velocity, the average pause time, the direction in which they're moving is sufficient to answer those questions. So even though the underlying situation is more complicated, we don't have to bother about that because we know they're manifest in the parameters that we're using. In the chromatin problem, by grouping huge 10 to the 6 base pairs into one unit, we're also making a comparable simplification. We're looking at the behavior on large scales, not on small scales, and that's a crucial thing. By looking at mesoscopic behavior and not at microscopic behavior, we can forget many of the detail that is important to the microscopics and concentrate on only the features that we are interested in. To give you another example, remember that even if we were trying to understand how water structures around the protein, we're not going to be modeling quarks and we're not going to be modeling what happens inside the nucleus. So we're already mentally making a separation of spatial scales over there, saying this is important, that is not important. But thanks for that question.
Okay, thanks a lot.
Yes, Professor Naveen Singh.
Yeah, Gautam, it was a nice talk as usual. So I have two small questions. So one is about this temperature you said in the chromatin problem.
Yeah.
So you put temperature in a reduced unit, or means you said 20 and 1. So is this on some scale, or?
When we simulate it, we simulate in reduced units. But we relate this to the numbers that are known for physiological temperatures, for the amount of energy that we release by ATP hydrolysis. So the simulation, as you know, it's always better to simulate in reduced units rather than actual numbers of 10 to the power minus 21 or so. So that's what we do. But we relate that to the real numbers underlying.
Okay. And second thing is just a small, I could not understand it. So in a cell, this broken road, how you can map broken road problem to the cellular environment? Because cellular environment, nothing is like a broken road. The picture you have shown, due to earthquake, we have a broken road. So how means we know that cell transportation, it happens that if there is any roadblock is there, it changes its path and try to move from other path, just like in crowded environment. So how you take care of that broken road problem into the cellular environment?
So that's exactly, so when it's known that if you look at cross sections of the axon, it's known that there are several breaks. We know the density of breaks along the axon. And these are points where the microtubule is broken. So actually, it very literally is like a broken road. We do exactly what you suggest. We allow the vesicle in the motor carrying it to be able to sidestep, to move to another road to the side or here. We know a little bit about the rates at which that happens. Because it's very dense, we don't think that coming off and reattaching somewhere else is an important point. Normally, you would have thought that that was the important thing, but this is so dense. This particular neuron is so densely packed. The axon in this neuron is so densely packed that even if it comes off, it will pretty much reattach very close to where it came off. So we argue that to first approximation, you can neglect that, but you cannot neglect the moving side to side to another track on the right or the left, or moving backward. These are what we consider to be the important features of that particular description.
Okay. Thanks.
Thank you.
Professor Venkatesh. I've seen his hand raised, so I probably by mistake. Yes, anybody else who wants to ask? Okay, Professor Jayendra.
Hi, Gautam. Very nice talk.
Thank you. Hi, Jayendra.
Hey. So I just wanted to not about your talk because I know you are working on that COVID-related thing. I was just looking at some of the papers that recently you are also written one paper on vaccine allocation strategies.
Yeah.
So can you just say about the strategy? What is that you are trying to say there?
Oh, lovely question. In fact, I thought maybe I would talk about that also here. as a real example of it. I didn't think finally too many things in a talk is probably not a good idea.
Yeah.
So I can tell you what the issue there is. So you know that when India does testing, when we do testing in India, we do a combination of two tests. We do the RT-PCR test and we do the rapid antigen test.
Yeah.
The RT-PCR test is very accurate, but often the problem is that the results are delayed. Right now in Delhi and other cities, they're being delayed by 4 to 5 days if you want to get a test. And on the other hand, they're also more expensive tests. Right now, they started around 2,500 rupees. Now, because government has beaten the manufacturers down, it's about 700, 800, which is pretty much a low cost given the cost of inputs that go into that. The rapid antigen test is less sensitive. It's probably sensitivity is 60 to 70%. But the advantage is that it's an immediate test. You can get the results in 15 minutes. And it's also a cheaper test. So the question that we wanted to, so if you look across India, the state of UP, the state of Bihar were all using 60% to 80% rapid antigen tests and not PCR. So many people shouted at them saying that, look, why are you doing this? You know that the RAT tests are not sensitive tests. You should always do the PCR, etc. But the governments, of course, are in a quandary because they're spending 2 or 3 times more to do a PCR test than they do a rapid antigen test. So then the question is, what is the trade-off? How many rapid antigen tests should you do versus PCR tests at a given sensitivity to achieve the same results, that catch the same number of cases that you might do in one case over the other? So that work does a network model where it simulates spreading on a network. You have people at home, people at work, people in hospitals, people moving backward and forward between these. It sort of examines various strategies. The moment someone tests positive, you quarantine them, you quarantine the entire family around them. And then it asks, if I have different levels of RATs, 20% RAT, 80% PCR, 50% RAT, 50% PCR, and different regimes, it asks, what is the advantage that you might get? How bad is the use of RATs? So our final result, which is really the important result for policy, is that it doesn't really matter. You can even have 50, 70, 80% of RAT if you test more. If you test about 4 to 5 times more than you're currently doing, you can get by with the use of RATs, and it's also economically more sensible because the difference between the cost of the RAT and PCR is such that there is a certain cost-benefit ratio that is involved. So as a function of the test sensitivity, how many tests you do per day, as well as the cost of the 2 different tests, you can figure out where you should be, where the state government should be making its decisions. So that's, I think, is a nice example of how mathematical modeling leads to very concrete suggestions for policy. That's something that is of particular interest to me in all of the work that I do nowadays for COVID-19. How can we make better policy that saves more people?
And okay, maybe I just another question I ask related to this.
Sure.
Now, where we are moving, I mean, this rate is increasing. What do you think that? Okay, and what is your model is telling?
So the models that I look at say that the rise in cases will go on at least for another 2 to 3 weeks. They may come down earlier in Maharashtra compared to the rest of the country. The rest of the country will probably go on to see a peak at least maybe 6 weeks from now. 4 to 6 weeks from now would be a guess for that. The problem is that we don't know many of the details regarding what is happening right now. One idea, the idea that I believe is that the new rise in cases comes from new variants. So something has changed from last year to this year. The second wave is being really driven by these new variants that have come in. These new variants are more transmissible between people. So you hear many more stories of whole families catching it from one infected person. Whether they lead to more mortality is not clear. Maybe they don't. But also the important thing is to what extent are they leading to reinfections? That people who've already caught the earlier strain of virus, are they more affected now? Are they completely safe now? To what extent, if you're vaccinated, are you safe now from the new strain? Yeah. We don't know any of these details. We're waiting for government to tell us these details. Because then we can put them into models. If we don't have these details, the model predictions will change. So currently, we're not putting any of this in. We're assuming that if you were infected once, you will not catch the infection again. And that leads us to these numbers. But that may not be true. You have to be careful and see what happens in coming days.
So that prediction may change whenever you will have the data.
You should never really believe any prediction more than 10 days to 2 weeks in advance. So in even what I have told you, you should not believe. I think cases will go up for the next 10 days to 2 weeks. That is more or less my prediction.
But ICMR probably
is hard.
Okay, but ICMR probably access of all this latest data. So
ICMR is not a transparent organization which is making this data available. We would like to know, they have access to all the data that will tell you what is the fraction of reinfection, because they have a unique patient ID. They know when the patient first tests positive, they know when they tested positive again. Combining that with genomic information as to what is the strain that infected, what is the variant that infected them the first time and the second time, will really give you a correlation. Are you seeing people who are getting reinfected because they were reinfected with a new strain? Can we ignore reinfections completely? What is the extent to which the new strains are spreading across India? Is it driven by strains, or is it driven by just public behavior? Just more people going out, Kumbh Mela, elections, all of this. We have to refine all of our models to deal with these things. We're currently trying to do all of that.
Okay. Thanks a lot.
Yeah.
Okay, we can take last question from Nikhil.
Sir, I have a couple of questions. In your earlier study, what was the cargo in the neurons?
So the cargo is a bunch of protein molecules that are intended for the synapse, right at the end of the axon. So we know what those cargo molecules are. We know what they're typically neurotransmitters that have to be moved from here to here. And what is moving back is typically disassembled motors at the end of that, various things that you break up and then you take it back to the cell body so they can be recycled. So we know what is the nature of the cargo. We can label specific cargoes inside here and look at them as they're being transported back and forth. But largely neurotransmitters would be the answer.
So the only the electricity is being transmitted at the synapse and not a physical molecule.
No. The electricity that is transferred is an instruction to the synapse to release the vesicles that have come that are present there. So you pile up, all the vesicles are piled up here. The moment it receives an instruction, which is an electrical signal that passes, these synapses fuse with the membrane of the cell and they're released into the space in between cells. Because these cells are so close to each other, the release of molecules into the space in between means that they go and bind to the cell in front of them. That's how a signal is propagated from neuron to neuron. It's the combination of an electrical signal, the release of neurotransmitters, the stimulation of the cell in front of you, and again this proceeds across these different cells. And as these proceed, they can the contacts between cells increases or changes or can be strengthened or weakened. And this is how memories are formed. That's our current understanding.
Okay, so you said that the microtubules break and form often, and why aren't they permanent if the transport is happening all the time? Why aren't they permanent?
So that's a good question. It's not, so in the cell, they're more permanent. The picture that I showed you of the thing opening up and closing is sort of more seen outside the cell. There are breaks. And at those breaks, there are regions where you have other proteins that come and bind to protect those breaks, etc. But all biological polymers do polymerize and depolymerize. It happens all the time. It happens in multiple contexts. When cells divide, they need to break up all the polymers, make them back monomers again, help the cell divide, and then reconstitute all of these polymers again. So whenever you have molecules, this is just biochemistry. They keep extending, shrinking, extending, shrinking. There's nothing to say that they have to have a fixed size. So this is completely part of chemistry and biology. So this is very natural.
Okay, and the microtubules are hollow. So could the internal part of the microtubule also be used for motor transport? Do we know that?
No. The motor plus cargo is much bigger than it's about 24 nanometers in diameter. That's not sufficient for a motor to be able to carry cargo. What is exactly happening inside is not very clear. It's idealized and pictured as a hollow tube. Whether there are small protein molecules that sit on the inside of those tubes and do something, we don't know. But it's believed very strongly that nothing happens on the inside. There is no transport that happens on the inside. It's always external to the hollow cylinder.
And is the ATP being absorbed at the motor by diffusion or something?
Yeah, it happens through diffusion. The ATP molecules are present in high density in the surrounding cytoplasm of the cell. They come and they bind to the motor domain. Once they bind to the motor, to the ATP binding domain, once they bind, they're broken up. The ATP becomes ADP. The ADP and the phosphate is released, and that energy is used to power the motor from moving forward. So how the ATP reaches is purely by diffusion. Good question.
And depending, as we know that when I touch a hot surface, I quickly back away. So that is the speed of the reaction. This is the reaction time of the muscles, the information being carried out. So I'm assuming it to be very fast, right? So
but those are sort of involuntary motions. So they don't involve this sort of transport of
the muscle memory part is something.
That's slightly different. But finally, this process of transport from nerve to nerve is very crucial. This is exactly how it happens. You have cargo moving from here to here, not just in axons, but also in regular cells that are not nerve cells. You have cargo moving back and forth from the center of the cell to the periphery of the cell to various organelles within the cell, and they're all being moved by these molecular motors. In some cases by diffusion, but in large cells, almost always through molecular motors.
Okay, so the quick reaction time is the signal is not coming to the nervous system, the brain. It is just up to few dendrons, and I'm backing off due to the muscle memory part.
You should read up that these involuntary sort of motions that you get from just touching something. These are sort of somewhat faster than the ones that are obtained by the slow motion of motors carrying cargo and depositing stuff at the synapse.
Okay, and are the motors unidirectional? Are they coming back? Are they reused?
Some motors are so every motor has a unique direction. So the kinesin motor moves from the cell body to the periphery of the cell. The dynein motor moves from the periphery of the cell to the cell body. Motor components are reused and recycled. Yes.
Okay. Okay, that's it. Thank you very much. I was impressed. The talk was very great.
Thank you.
So there will be no more question, and thank you very much for this excellent talk. We all are benefited by your talk, and we would be very happy to welcome you physically, and hope that we will have a corona-free time in near future.
I think we should all hope that. Yes.
Yeah, we hope for that, and we would be very happy to welcome you physically here.
I would be very happy to come. Thank you for the invitation, and all of you, please stay safe and stay healthy. These are all difficult times, and I look forward to seeing all of you sometime in the future.
Okay, thank you very much. And yeah, anybody wants to add anything?
No, it's thanks. And
okay, goodbye. Good night to everyone.
Bye. We can discuss now.